So very good afternoon, everyone. My name is Pavan Kumar, and I thank you all for joining this fourth part of this open webinar series from the IBDC. In this webinar, we will discuss about this macromolecular data portals which are developed at this IBDC. And during this presentation, I will go through a little bit briefly about the IBDC functionality, its application and usage, and then about this ISDA data portals, which is the macromolecular data portals, and then basic understanding about this macromolecular types and why it is important in research community, and what are the functionality and features is provided by these portals, as well as application and benefits user can use during this application, this data portal type. And during this presentation, your microphone will be automatically switch off. So please use this chat box for any questions, and our ISDA team will continuously reply to your question during this seminar. This recording will also be available at this IBDC website. So for any future communication, you can use this recording for review this recording. So start with this about this IBDC. So IBDC is the first national science data repository in India, which is supported by Government of India and through the Department of Biotechnology. And it implements this biotech pride guidelines. Any user interested about this guideline, you can check our website, IBDC website, or our previous webinar for more details. This IBDC is the joint project of RCB, NI, ICGB, and NIC. And at present, it is housed at the Regional Center of Biotechnology, and which is the UNESCO category second center. So at the heart of this IBDC, we have this high-end computational infrastructure, which is housed at this RCB. And we can also have this disaster management site at NIC Bhubaneswar for real-time data exchange, so that any casualty happen at the data center, we can take the backup at the real-time manner. And using this IBDC, this HPC, we are employing this IBDC portal, data submission or access dissemination of this data, and then curation of the data, primary archiving of the data, which are submitted at the IBDC. And this is our main building. And any visitor wanted to visit, you are happily visit this website, this building, and we will welcome with more detail about this IBDC functionality. So as we know about this data diversity within the biological samples. So data means coming from this gene genome to the functional genomics, proteomics, macromolecular structure type, metabolome type, or image data, medical or the clinical image data, as well as the phenomic data. So these are the diversity which mostly belongs to from the biological data type. And to capture this information, at present we have 7 different data portals, ranging from the nucleotide data portals, that is the INDA, then phenome data portals, ICPD, macromolecular structural data portals, that is the ISDA, metabolome data archive portal, IMDA, and then proteome data portal, IPD, and very recently we have released this biological image data archive, that is IBIA. So interested user, please check this respective website for more information. Now, coming to the macromolecular data portals, the topic of this webinar. So, and we will discuss this Indian Structural Data Archive, ISDA, then which are developed at this IBDC to analyze and then store the macromolecular data type in the IBDC. So why we need this 3D macromolecular structure information? So as we know, this biological macromolecules include this 3D structure of protein in connection with RNA, DNA, or the polysaccharides. And this macromolecular entity provide the information from the basic atomic level information to the large subunit and assembly informations. And this basic subunits serve the basic unit to understand the macromolecular level at the functional dynamics. So just to empathize the role of this macromolecule in the context of the protein structure, so having this macromolecular structure, so we can identify the new folds, we can analyze these protein-protein interactions, we can understand the protein-ligand interactions, we can understand the role of the mutation over the protein 3D structures if any mutation occurs, and then at the last means protein dynamic interactions. So how these proteins behave over the dynamics range. So these are the major applications. If we include this protein interaction in context to RNA, DNA, or polysaccharide, its range will further increase. So before going to the means actual this data type, so why we it is important to understand why we need this WWPDB and its requirement. So the worldwide PDB organization which manages the PDB archive and ensure that this PDB this data type is freely and publicly available to all the users along the means across the global community. So it implements this FAIR principle to this archive this data type. It provides this deposition and validations and biocuration activity at no charge to the all means worldwide to worldwide depositor, and also ensure this open data access to the public domain to all the structured biological community with no limitation on usage also. So you can use as many times you want to use it. And this also developed this community endorsed data standard for the archiving and the exchanging of the data globally. And from the figure, you can understand how this WWPDB this organization associate with the 4 different members to real-time synchronization of the data and maintaining the repository. So coming to the portal type, that is the Indian Structural Data Archive. This Indian Structural Data Archive is the comprehensive repository which has 3D biological macromolecular structure informations that we weekly synchronize all the updates from the WWPDB in the database, in the ISDA database. We also curate Indian origin macromolecular entry and distribution. It's along the statewide distribution. Then we curate the entry and update the Indian entry to the all the users of the ISDA. And along with this curation, we also try to provide the functional insights which are coming from this biological structure, macromolecular structure, or the biological interaction type. So user can visit this ISDA data website using this ibdc.dbt-india.gov.in/ISDA. So just an overview of how we are this archiving this data in the ISDA. So on the weekly basis, we get all the latest update information in the form of MMCIF file format, PDB file format, XML file format, and a small micromolecular dictionary, that is the CCD formats, to extract different level information like this experimental detail information, citation detail information, micromolecular detail entity information, as well as small molecular entity informations. All this information we weekly update from this WWPDB in our ISDA database collections. After getting this stage collection, we get the after getting this first stage information from this WWPDB, we annotate this all the PDB entry in terms of scope, cat, or the geo, or the uniport ID, or the entries ID, so that it's gets more relevance in terms of functional annotations. Then we, at the next stage, we put more interaction and assembly analysis information. We provide this this protein structure image, protein ligand, protein interaction image, as well as ligand image for more visual interactions at the portal level. And all this information passes to the ISDA database, and so that the user can get the all the real this information and from the one space. And to serve this backend, this all the information to the user, we employed this Node.js and GraphQL and MongoDB database to store all the SDA information. No, it's very relevant to when we have a large number of field to search, so that we can search means explore the database and very different perspective. So at present we have around more than 40 different search fields, and for just to visualize here, we have categorized this all the 40 different fields in 5 different this section, like this PDB entry details. So user can use this organism name or the taxon ID or the gene name, Uniport ID to get the PDB entry details information, like or polypeptide information. For that, user can use macromolecular type or compound name, formula name, and functional annotation. For that, the user can use this feature type or the enzyme name, macromolecular name, classification known, then entity type or the cath ID, PFAM ID, scope ID. That information you can use to get the functional annotated information from the PDB, sorry, in this ISDA. Then user can also use this author to get the author specific search. So user can use this author name, ORCHID ID, journal name, DOI or the PubMed ID to retrieve the specific PDB ID from the ISDA database. We have to improve this search space in the ISDA. We have also have 5 predefined fields to make it more focused over this search and to reduce the search time as well as the accuracy. And we have this search filter so that user can further refine the search outcomes from the SDA, this search space. So this is this page. When you search over this ISDA, so these are the 5 different information where user can predefine search information. User can select and search in the database. And if you are not selecting anything, then it will be the free space search, and you will get all the relevant information matching to that query word. So this is the search result. And in this search result, the user will get this PDB, this entity image, as well as basic information about this classification type, release date, method used to resolve this structure, then organism belong to this protein, and then if any bound ligand molecule, then it will also highlight the this chemical ID. If and at the left side, user can use this refine space to further refine the query. Let's say you search for the transferase, and you want to only search over this Homo sapiens, then select over the Homo sapiens and click over the search so that you can refine further to the this transferase enzyme over the only belong to the Homo sapiens organisms. So like that, user can further refine this search result from the ISDA database. So when user click over this view button to get the more details, though, so detail page will open. So and from that there is a first page will be the PDB entry summary page. So in 4 categorical information you will get. One is the biological detail, second one is the experimental detail, third one is the citation detail about this published information in the journal, and then macromolecular entity as well as chemical entity. So macromolecular entity will means how many chains are is there, description about these chains, length of the chains, and then it's annotated to Uniport entry. Then you will get the macromolecular information from this. And if any bound ligand is there, then you will get the chemical ID information, the chain it has bound, and then name of the chemical, and then 2D image of this image of the bound ligand. In the second page, you will get the more detail about this 3D visualization of this protein, as well as sequence completeness, means whether it is complete sequence, which is referred to the Uniport sequence or not, and then functional annotation, which in terms of cath, geo, and then PFAM ID, etc. Next page highlight the experimental details about this protein, which are used to crystallize or to resolve the structures. So this is information means related to all the experimental details of the protein. Now, if your protein entry is having a ligand molecule bound with this within the PDB entry, then user can get all the information ranging from this 1D information in terms of SMILE and key, and then this 2D image information to depict the 2D structure. And if you want to see the 3D structure, click over the 3D view so that you can check the 3D information of the protein, this bound ligand information in the different browser. Along with the all the information coming from this WWPDB, we are also curating this Indian origin structural records so that we can highlight how many structures are coming on the weekly basis from the Indian community, and what sort of information we will get if we use this graph stat, so that we can check which proteins Indian structural community is working more towards like this. User can check the source organism. User can check the protein type, experimental type, mostly used by this Indian structural biologist, and then year-wise distribution. Sorry. So that it will clearly reflect the type of information and this growth of this Indian entry along the this time. So we have also have a different level of this curated pages, so that user can get the relevant information from the single click. Like we have COVID-19 page, so all the protein information along with this reference ID and this chain length and the description, as well as the number of structure belong to the particular protein is user can get from the one page. And it is also further segregated over the method type which is used to resolve the structure. So all the information we are curating in the one place, so that user can get the real-time update about the any proteins which are belong to the COVID-19 virus. So that which also highlight the the next stage is the let's say we classified all the information at the taxonomic divisions. So 5 major taxonomic divisions we have classified, and user can get all the information in terms of PDB structures from this any division virus belong to the 5 different divisions. And if you view over the more button, then you will get the whole list of this virus or any taxonomic divisions, and user can get how many structure belong to the particular organisms. And if you click over that, then it will move to the search space, so that you can get the all information along with the image and the other details from the search page. We have also provided the curated collections about this protein oligomeric states. So how many entries are the single mer, or the homomer, or the heteromers, so that user can get all the heteromers in the one click. Or we have this enzyme classification over the all the classification, so that user can get which protein enzyme type is mostly resolved in the PDB structures. And these are the type 5 protein structure by the PDB entry, so that user get all the information about this top 5 most this crystallized PDB structures. Then the more important this functionality that we have developed is to get all the information which is based on this number of amino acids bound to the particular PDB entry. So let's say we are searching for the any protein which having only the 10 amino acids long sequence. So clicking over this one portion, user can get the only entry which having the all 10 amino acids long chain within the PDB entry. So that's how this functionality we have developed, so that user can get the one-click solution to the relevant this question. Now, apart from this experimental entry, we also have this AlphaFold model structures, and this at present we have more than 8 lakhs AlphaFold protein model structures, which includes more than 5,000 organisms and more than 10 model organisms And at present we are more focusing over this insect organism so that we can serve to the Indian scientific community to work over the insect, which to design the more accurate this pesticide to serve for this agriculture and the crop biology. So user can check all the structures which are listed for this AlphaFold classification, and you can search. And if you have any more this organism name you want to list and this list, so that you will mail us at the support@ibdc.rcb.ac.in so that we can design the model the structure and listed here. Now, as this is my pleasure to inform this to share the news that now the transition is moving from this structural biology to the computational biology and AI, which are helping us to design the protein of choice as well as the protein 3D structure, which was nearly a decade-long open challenge within the scientific community, and very recently this Nobel Prize was given to this chemistry 2024 for this protein design as well as protein structure prediction. And which is the second one part is given to this David Baker, and so second part is given to the this Google DeepMind this team to design the AlphaFold model solution to predict the very accurate protein structure to serve the community. Along with this for the experimental structure information, we have very real-time stats about this PDB entry, which is user can check to see which information is growing, and which means let's say if we look at this resolution by distribution, you can check how many structure are populated over the different resolution range. User can also check how many number of chains are there with respect to this protein entry within the PDB structure, and then how this three different major and this experimental type like this X-ray, NMR, and then EM are growing with respect to the time, and how the number are populating in the PDB structure. And in the general scale, you can check what are the preferred journal type which the scientific community prefer to publish their work, and what are the major source organism from which proteins are extracted and crystallized and submitted in the PDB structures. So these are the stats, real-time stats the user can check to see the growing aspect of the PDB structure. Now, apart from this this ISD, we are also adding this some more information about this emerging pathogens dedicated pairs, and in which we will group this experimental and the computational model structure at the protein-wise level across the genome. So like this we have done in this COVID-19, so that all the emerging pathogen, you will get the very specialized solution in terms of number of structures available across the experimental as well as the computational model source. And we will also provide this protein instruction information within the page, so that user will get the protein instructions or protein ligand specialized instructions at the one place. We will add this genetic and somatic mutations to connect all the information to the disease mechanism. So we will connect the disease biology also in that page, and we will involve the pathway information, so that user can see what are the pathway are enriching if you selecting the particular protein type to understand the disease mechanism. We are also connect to this page information to the chemical information to drive this computer aided drug design process. So this will enhance and then enrich the all sort of information which are emerging related to the emerging pathogen within the India. Now, we are also developing one more this analysis toolbox within for this all the user base so that the user can get a dedicated dashboard for functionality to analyze the genomic data type, macromolecular data type, or the biomanufacturing data types. And it will be powered by the IBDC Brahm HPC. So you will get all the resource map at the behind to analyze your data type. User will be able to design this analysis pipeline as per the requirement. So in the coming month, please check out our IBDC website so that you will get to notified about this release of this analysis toolbox from the IBDC website. So at the last, this is the team which is working with this ISDA. So Himanshu is this database curator, and Mayank and this Kalpana is the software developer, and me is the Pavan Kumar is also a database data curator. I want to acknowledge our head, Deepak T. Nair, and also core committee members, and the Department of Biotechnology for funding these projects, and this IBDC and the RCB for all the resources. I also want to acknowledge this data resource from this WPDB and AlphaForce for make it available for all the user community to enhance the scientific understanding about the 3D macromolecular data. And if you have any query, please send us to support@ibdc.rcb.res.in. Now we are open for all the question answer. If you have, please write in the text box. In the future, if you have any question, please write to us at support@ibdc.rcb.res.in for further any knowledge you want to get from the IBDC.